All right. So what we're going to do today is to gel some multi-domain peptide with HBSS. Now, the multi-domain peptides, the ones that I'm going to talk about today, have terminal lysine residues, which can be cross-linked. The charges can be shielded, and the peptide can be cross-linked with the addition of multivalent anions, such as phosphate, which is present in PBS or HBSS or what have you. Now, typically, I wouldn't use media, quiescent media, without the addition of BSA or FBS rather, is okay to use, but FBS or serum in general, or BSA for that matter, precipitates out the protein. So I'd avoid using that. For the example today, what we're going to do is gel the multi-domain peptides with HBSS. There are two peptides we'll be working with today. One is called SLANC, S-L-A-N-C, and by visual inspection you can see that SLANC is not that viscous compared to another peptide called SLAC, S-L-A-C, which is a lot more viscous, and you can see that it's really viscous. You can see how it's flowing there, kind of like honey or molasses. Now, for the purposes of this demonstration, what I'm going to do is I'm going to fill this 16 well chamber slide. I'm going to fill 1 or 2 wells with each of the peptides to give you an idea of what it should look like. Maybe I'll just do one of the peptides. Okay, so the first thing you want to do is to have your buffer solutions ready and aspirate, or rather pipette, how much you need. So let me just get a pipetter. Sorry. Mind if I just get behind you? Okay, there we go. I would add the buffer solution, such as HBSS, phosphate buffer, or what have you, or even media without serum, to an Eppendorf tube, the microcentrifuge tube, ahead of time. So if you want to make 50 microliters of gel, I would suggest making at least 75 to 100 microliters. I would always budget 50 to 100 microliters extra to make sure, because of viscosity, that you have enough gel. So what we're going to do is we're going to take a pipette tip. We're going to draw up the HBSS solution first. So I'm going to take 50 microliters, draw it up, and you can see it's inviscid, so it aspirates very easily. I'm going to put that into here. Now, it goes without saying, definitely want to use a new pipette tip for the solution, especially because the HBSS, while it's sterile, will cross-link the peptide if it's added to it. Now I'm going to work with the more viscous peptide. The less viscous peptide, SLANC, you could probably just aspirate that pretty easily. The more viscous peptide, SLAC, SLAC, what I'm going to do is I'm going to set the pipetter to about 25 microliters above what I need. So in this case, I'm aspirating 50 microliters, so I go to 75. If I was aspirating 150 microliters, I go to 175, for example. So I'm going to go ahead and depress my pipetter, go into the solution, and I'm trying to avoid the bubbles. If there are bubbles, you can use a centrifuge to get rid of the bubbles. These are resistant centrifuging. You can centrifuge this all day long. So I'm going to slowly aspirate this, and as you can see, I'm trying to avoid the bubbles, as I hope you can see. I'm bringing it into I'm just leaving it in the solution for a good 5 or 10 seconds to let it equilibrate. And after that, what I'm going to do is I'm going to go ahead and turn down the pipetter to 50 microliters, which is what I need. And any excess which has gone into the pipette tip is now going to start coming out. What I'm also doing is ensuring that when I pull out the pipette tip that there isn't any air that goes in, suggesting that we didn't get all the volume that we needed. What I'm going to do now is to go ahead and put the peptide inside the HBSS, rather the pipette tip, and slowly push out the peptide. Now, if you had a close-up view or a good enough view, you can see that it forms like the strand-like material. So all I'm going to do is just basically pipette up and down to give it a good mix. And what you can do at this stage is actually take this, put this on a vortexer, and that will allow you to mix it really well. And as you can see, it still flows a little bit, so you can still aspirate this. So you can go ahead and put it on a vortexer, vortex it, then centrifuge it to remove bubbles, and then go ahead and put it in your microwell plate. In the event that you have cells within the HBSS or cells within media, I would definitely recommend against vortexing and centrifuging. So just be careful not to get any bubbles. In this example, I'm just going to go ahead and tap it on the bottom of the cell culture hood, and that kind of gets out any of the bigger bubbles which are in the solution. What I'm going to do now is to set my pipetter to about maybe 50, maybe around 60 microliters. I'm going to pipette up the peptide solution. If I wanted 50, I set it to 60. Pipette up the peptide solution. So right now the peptide solution is a hydrogel. The initial peptide is not cross-linked, now the peptide is cross-linked into a hydrogel. But you can see that it still aspirates, and that's one of the nice things about this peptide system. So I'm going to drop it back down to 50, which is how much I want to put into one of these wells. And what I'm going to do is to go ahead and just go to the corner of one of these wells and put it in. Now I got a bubble there that's obviously not good. I want to avoid that, and I can do it that way. You can tap the well. I mean, whatever technique works best should be just fine. Typically for these, maybe 100 microliters works better than 50, but you know, it's just a demonstration. Okay. Now typically what I do is I would take this, obviously label it, set it in the incubator at 37 degrees centigrade, 5% CO2, in a regular cell culture incubator with humidity, and let that sit for 30 minutes before I added buffer on top. But since, again, I'm just doing this for demonstration's sake, what I'm going to do, and by that time, the hydrogel should be pretty stable and resistant to any shearing due to media you put on top. But in this case, just for demonstration's sake, I'm going to go ahead and take 50 microliters of quiescent media. The media that you put on top can be anything because you're not gelling the peptide with it. You're just adding the media for the betterment of the cells. And what I typically do is that I just drip it on the side of the wall above the hydrogel. And if you get it just right, what you'll see is that there's a layer of hydrogel below and there's a layer of media on top. Now in this example, I've used very little hydrogel, very little media. I'm just trying to conserve a little bit. But what you would see is that a clear, distinct, clear layer of hydrogel from the bottom to a certain height, and then media on top. And that's pretty much it. And you can let that culture as you would. One of the things with our particular peptide amphiphile systems, with our peptide systems, is that they start to take up indicators. So what you might notice in a setup just like this is that on day 2, your hydrogels might be a little yellow, for example. They take up some indicator, and small changes in pH can cause the indicator to change color. But obviously, you know, a regular cell culture procedures from here on. So that's the general principle behind gelling with these peptides. So just keep in mind, you can always add multivalent salts. I typically add them in a 1-to-1 ratio volumetrically, so 50 microliters of this, 50 microliters of buffered solution. Just keep in mind the viscosity concerns. Feel free to go ahead after you mix the solutions together to vortex them, and then centrifuge to remove bubbles, and then aspirate carefully to avoid bubbles further still. But other than that, that's how you do it.